A-Level Biology 9700, Paper 4, Variant 2, February March 2026. And these are the two last questions on this paper, question number 9 and 10. So the question number 9 is there, and question number 9 is: nitric oxide is a cell-singling molecule that has a role in a number of physiological processes in the body. It is also used as the therapeutic drug. Nitric acid is known to have harmful effects in body cells if conditions are not controlled. Scientists investigated the effect of nitric oxide on the use of oxygen by kidney cells grown in a culture medium. The scientists recorded the oxygen concentration in the culture medium over a period of 24 minutes. During this 24-minute period, a small volume of nitric oxide was added to the culture medium on 3 occasions. The results are shown in figure 9.1. So here you can see the time has been given on x-axis, and on y-axis oxygen concentration in culture medium arbitrary units has been given. So here you can see that the oxygen concentration is decreasing as the nitric oxide is added into it. So from 0 to 24 minutes, it becomes around 0. So describe the results of the investigation and suggest an explanation for these results. So here you can see in this question that the overall trend is that the concentration of oxygen decreases over time uh, from 15 arbitrary units at 0 minutes to 1 arbitrary units at 24 minutes. So little bit more than the 0. So then you can see the effect of nitric oxide as the time nitric oxide is added at, first of all, it is at 3. You can see it is at about 3, and then and at 8 minutes you can see, and then at 15 minutes, and then the rate of oxygen consumption then at 20, and it is not adding at 20, so it is only at 3, and then it is at 8, and at 15. So you can see as the rate of oxygen consumption decreases as the nitric oxide is added into it. So before each addition, you can see that the oxygen consumption resumes and the rate of oxygen decreases different again. You can see this. You can see it try to resume, but when we add the nitric acid into it, then it starts decreasing again. So I'm going to write all this description about results over here. I would like to write over here as the decreases over time from 15 arbitrary units at 0 to 1 arbitrary units at 24 minutes. Each time when nitric acid is added, the rate of oxygen consumption decreases bit before each addition of nitric acid. The oxygen consumption resumes and the rate of oxygen and the rate of oxygen decrease sequentially. Nitric acid acts as a inhibitor because over here the oxygen is used as the final electron acceptor in the mitochondria. This slows down or halts the electron transport chain. You can see the oxygen drops from oxygen drops from 15 to 11.2 arbitrary units, and first 3 minutes. Next part of this question is B. Some organisms are able to carry out respiration in anaerobic conditions. Describe the differences in respiration in anaerobic conditions between mammalian muscles cells and yeast cells. So here you can see that we have to write down the or describe the differences in anaerobic respiration between mammalian muscles and yeast. So first of all, you can see that the in mammalian muscle cells and yeast cells, when we talk about the mammalian cells, the end product is the lactate or lactic acid, whereas in yeast cells, the end product is carbon dioxide and ethanol. And the enzymes which are involved in both of these type of processes, in mammalian cells, the enzymes are both enzymes are different, and the same is, you can say, the in the first step pathway in the mammalian muscle cells, from pyruvate to reversible lactate can be converted back to pyruvate in the liver, whereas the yeast cell, the pyruvate which is formed, it is not irreversible because the carbon dioxide is lost and there is no formation of carboxylation or the formation of carbon dioxide. Whereas in the other, when the yeast, the carbon dioxide is produced. So these are the major differences between both of these processes. First of all, I would like to write, or I would like to compare both of these processes as the mammalian muscle cells and product is lactic acid or lactate. Whereas in yeast cell, the end product is carbon dioxide and ethanol. Enzyme lactate dehydrogenase is involved in muscle cells and in yeast cell. Alcohol dehydrogenase. Nase. The muscle cell reaction is reversible. Reversible, that is, lactate can be converted back. can be converted back to pyruvate. Whereas in yeast cell, the reaction is irreversible. O carboxylation no decarboxylation. Let me write it again. Decarboxylation occur in muscle cell, whereas decarboxylation, carbon dioxide is released in the first step. Next question. C. Rice plants are adapted to grow in flooded fields. Figure 9.2 shows rice plants growing in a flooded field. So explain three adaptations of rice plants and allow the rice plants to grow in flooded fields. So you know that the flooded fields where the you can say the rice plants are grown, the presence of iron gamma tissues is there. That is, the iron gamma cells are those cells, you can say, which have the air channels in stems and roots, which allow the oxygen diffusion. And then it has high tolerance of alcohol or ethanol. And then you can say the stems grow very rapidly, and the shoots are submerged, they are elongated shoots. So in this way they are, and sometimes I would like to say that they leaves are traps a thin layer of air around submerged leaves to facilitate gas exchange underwater. So these are the different, you can see the adaptations. First adaptation is, that is, they have parenchyma tissues. Thus provides air channels in stems and roots, allowing oxygen to diffuse from aerial parts to submerged roots. Submerged roots rely on alcoholic fermentation. alcoholic fermentation, rice cells produce higher levels of alcohol dehydrogenase to break down ethanol. Internode growth becomes rapid. So leaves and flowers. So leaves and flowers stay above the water level for gas exchange and photosynthesis due to shoot elongation. Rigid leaf 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 surfaces traps a thin layer of air around submerged leaves to facilitate gas exchange underwater. So it was all about the question number 9. So moving towards the question number 10. So question number 10 is stomata have an important role in photosynthesis and transpiration. Figure 10.1 shows the formula for calculating the stomatal index for an area of leaf. Stomatal index is equal to number of stomata divided by number of stomata plus number of epidermal cells multiplied by 100. So this is the formula which we have to use to find out the missing values in table 10.1. So complete table 10.1 for cotton and tomato using this formula. So first of all, we have to find out the cotton. Cotton, they have given the number of stomata per unit area. Then we have to find out the number of epidermal cells. And then the stomatal index has been also given. So by using this formula, we are going to do this question. So first of all, the stomata index for cotton. It will be for cotton. So stomatal index is equal to number of stomata divided by number of stomata plus epidermal cells multiplied by 100. So we'll put the values over here. So here you can say the stomatal index is 15.8 is equal to the number of stomatas. So the number of stomatas are 6 and number of stomatas are 6 plus we have to find out the number of epidermal cells. So the number of epidermal cells, we can put a question mark over it and multiply it by 100. So according to the mathematical calculation, we are going as cross multiplication. So it will be 6 plus E or number of stomatal. So it will be 6 multiplied by 15, 8. So it will be multiplied by 100. You can calculate it by yourself. So 6 plus E is equal to 615.8. Then E is equal to 37.97 minus 6 and epidermis, it is nearly equal to 31.97 and we can write 32 over here. Now the other value which we have to find out, that is the for tomato. And we have to find out the stomatal index. So the stomatal index we can write it as C. Stomatal index is equal to number of stomata. So the number of stomatas are 7 and then number of stomata plus number of epiderm, it is 25 multiplied by 100. So stomata, it will be 7 and you can use your own calculator. It is nearly equal to 21.875 or you can write it as nearly equal to 22. So these are the values which you can calculate by yourself because they are Very simple values which you can do. So we are moving towards the next part of this question after calculating it. Or you can also write it as 21.88. Next question is, the Russian wormwood Artemisia gimmelini has a stomatal index of 13.8 for its lower leaf surface and 1.7 for its upper leaf surface. Explain the advantages to the plant for the difference in stomatal index values. So here I would like to write the answer as what are the advantages? The lower epidermal dermis receives direct sunlight and heat less so, keeping temperature lower and reducing water loss via evaporation. And there will be the, you can say it will be easy for maintenance of gas exchange because it allows sufficient intake of carbon dioxide for photosynthesis while conserving water, especially during hot or high light conditions. So these two advantages can be written over here as I am going to write the lower epidermis receives direct direct sunlight and heat less, keeping temperature lower and reducing water loss via evaporation. Second can be written as allows sufficient intake of carbon dioxide for photosynthesis while conserving water, especially during hot or high light conditions. So it was the A part. Now we are moving to part B. Figure 10.2 is a graph showing changes over a 24-hour period in the carbon dioxide concentration of the air in a glass house containing plants. The glass house did not have artificial lighting, so the only source of light for the plants was sunlight. The carbon dioxide concentration in the external atmosphere over the 24-hour period is also shown. The air in the glass house was not completely sealed off from the external atmosphere. So any difference in the carbon dioxide concentration between the glass house and external atmosphere will gradually decrease unless there is a process of to maintain the difference. So on x-axis, they have given the time of the day, and on y-axis, percentage carbon dioxide concentration, which is first decreasing at 6. It is start decreasing, and at it is decreased very less than 0.02% at 10, and it maintains the same at 18 hours. It start to increase again in the glass house, and whereas in external environment it remains the same, that is 0.04%. So suggest how different limiting factors affect the rate of photosynthesis of the plant in the glasshouse over 24-hour period in figure 10.2. Moving towards the figure, you can see that at 10 to 18 carbon dioxide reaches very low. So all this which I have explained, I would like to write over here in the given space. You can see we will start at 6 hour to 10 hour. It is morning. So the light intensity increases. Increases, making light the primary limiting factor. As light increases, the rate of photosynthesis increases, leading to a steep drop in carbon dioxide concentration inside the glasshouse as carbon dioxide is consumed. At 10 to hours to 18 daytime, you can see that the carbon dioxide reaches a very low level. Means maximum rate of photosynthesis. And then 18 hour to 22 hour the light intensity decreases. Decreases as sunlight fades, becoming the limiting factor again. Then at 22 to 6, which can be the night, completely stops photosynthesis. Completely stops photosynthesis. Respiration continues. Causing carbon dioxide levels in glasshouse to remain high. So it was all about the B part. Now the last part is C. ATP and reduced NADP are products of the light independent stage of photosynthesis state where in the chloroplast ATP and reduced NADP are produced, and describe the roles of ATP and NADP in the light independent stage of the photosynthesis. Well, the production or location of the roles of ATP is, you can say, they are produced in thylakoid membranes or grana of the chloroplast during the light dependent stage. ATP and reduced NADP produced in the thylakoid membranes then of the chloroplast during the light dependent stage. When the light is necessary, so the first part of the, or you can say in the light reaction. So the role of ATP in the light independent stage is role of ATP is in the light independent stage or in the dark reaction. Stage is to provide energy to provide energy to convert glycerate 3-phosphate into triose phosphate. And provides energy for the regeneration of RUBP, which is ribulose biphosphate from triose phosphate. Then role of reduced NADP is to and role of reduced NADP is to provide hydrogen atoms to reduce glycerate phosphate to triose phosphate. So it was all about this paper, 42 February March 2026. Now please like, share and subscribe my channel to watch more videos on your papers, on past papers. Thanks for watching.